Okay, so given that you guys have started working on your potato diffusion lab, I wanted to give you guys some background on what diffusion is and how it works so you can start thinking through what happened with those potato cores. Again, we've missed some time with snow days, so things are a little out of whack, but hopefully by watching this video presentation, you can get the basics down and start kind of critically thinking through what happened there. So, to put it in its most basic form, the term diffusion just refers to when you have a lot of something in one place and not very much of something in another place, or maybe not any of something in another place. And those particles are going to move from where there's a lot of them, all crowded together, they're going to naturally spread out to where there's less of them. So we call that diffusion. We refer to it as, we'll say, the place where there's a lot of things piled up. So over here, we've got these green dots, and there's a ton of them all crowded together. That means we call that a high concentration of those dots. There's a lot of them in a small amount of space, so they're highly concentrated. Over here, we don't see any green dots. So this is actually a zero concentration, but we're referring to it as a low concentration over here. These green dots are what we refer to as a solute. That's the word here, solute. And a solute is anything that you dissolve into water. So you can take table salt, you can take sugar, anything that will dissolve into water. It basically looks like it disappears when you mix it in. That's making a solution. The solution is the whole thing, the whole liquid with the thing dissolved into it. The solute is what you dissolved into the water. So if you put sugar in your coffee, the sugar is the solute in the coffee. So in this case, we're showing these green dots as our solute. We have a high concentration of solute over here, a very low or zero concentration of solute over here. And so what you see in the middle is these solute particles are naturally spreading themselves out. And if you give them enough time, they'll basically get to the point where they're pretty much equal everywhere. Now, what we refer to this as, in a situation where you have a bunch of them piled up in one place and basically none of them in another place, that's what we call a concentration gradient. So you have one area where there's a high concentration, another where there's a low concentration, and then in the middle you have kind of a middle concentration. So you can see these arrows over here. We've got the big arrow, then the smaller, and then the smaller. When you have that situation of high concentration in one place, low concentration in another place, we define that as a concentration gradient. And anytime there's a concentration gradient, whatever those solutes are, these particles that we're talking about, they're going to experience a drive to spread out. They're naturally going to want to do that. So the next question is, anytime we're talking about diffusion, the first question I want you guys to ask yourselves is there a concentration gradient? So for that particular substance, is it piled up in one place and there's not very much of it in another place? If that's true, then you have what we define as a concentration gradient, and those particles are going to want to move and spread out until they're equal everywhere. But the second question you always have to ask yourself is, are they able to move? Even though they may feel that drive to spread out and make themselves equal everywhere, it's not always possible for those particles to get from one place to another. And if there's something in their way that they can't cross, then it doesn't matter how much they'd like to spread out, they're not going to be able to. So what we run into there is that a lot of times in biology, there is something in the way. It's generally what we refer to as a membrane. So a membrane is just a dividing barrier between two spaces. And they're generally quite thin, so it doesn't maybe look like much of a barrier, but for certain kinds of molecules and particles, it may be a substantial barrier. So the barrier we're usually talking about in biology is the membrane of the cell, because we're going to be talking about whether things can get into a cell or whether they can leave the cell. So we're going to talk about the membrane of the cell membrane. And that membrane is what we call a semi-permeable membrane. So membrane means a barrier of some kind. Semi-permeable means, so permeable means things can get through it. Semi means only some things can get through it. Now, we see things that act like semi-permeable membranes in our real lives every day. If you come up to a chain link fence that has no cuts in it, it hasn't been broken in any place, that's a barrier to you. Now, you might be able to climb over it, but you can't squeeze yourself through these spaces in the chain link fence. So for you, a chain link fence is a barrier. It might as well be a wall because you can't go through it. But if you're a little sparrow, like these little guys, a chain link fence is not a barrier to you at all because you're plenty small enough to squeeze through these holes. So your cell membrane acts basically just like that. There's certain things that will see that cell membrane and won't see it as a barrier at all. Maybe they're small enough or something about them makes it so that they can cross the cell membrane without any problems. But other things are going to come up to that cell membrane, and no matter how much they might like to cross it, they're not going to be able to. So a semi-permeable membrane is a membrane that will let certain things through, but not others. So when you're talking about diffusion, the first question to ask is, do I have a concentration gradient? So do I have something that would like to spread its way across that membrane? And then, if there is that membrane in the way, is the particle that I'm talking about physically able to cross it? Or is it just going to get bounced back off of that barrier? So when it comes to cell membranes, there's basically a couple of major things that determine whether you can get across it, like a little sparrow hopping through a chain link fence, or whether you get stuck and can't cross it, like a human trying to squeeze through a chain link fence. And those two things, the two things that we need to think about the most, are if a molecule wants to get across a cell membrane, so over here, this weird looking thing, this double layered thing, this is our cell membrane. We will talk about what the nature of it is and why it's structured the way it is more, but for now, for you just thinking about your lab, the main things you need to think about are, okay, here's my cell membrane, which means all of your potatoes have these kinds of membranes in them because they are made of cells. So we've got your cell membrane, and we've got certain things that can cross it easily, certain things that can cross it with difficulty, and then certain things that can't cross at all. So the two factors that are going to impact that are, if you're a molecule, how big are you, and what is your charge? So you can think of the cell membrane as having kind of a force field within it. And this middle part of the cell membrane is a force field that will reject anything that has a charge on it. So this force field area in the middle hates anything with a charge. If it's got a positive charge, a negative charge on it, it's going to get rejected. There's really no way for it to cross that force field. So anything coming up to a cell membrane that has a charge on it is going to get bounced off. It's not going to be able to cross that cell membrane. So one thing is you got to look and see if the substance you're looking at has a charge on it. If it does, it's not really going to be able to cross this membrane. We also have to look at the size of the molecule. So if you look at glucose and sucrose, those are forms of sugar, very common throughout the body, but they are very large molecules. So it's not that they have a charge on them. That's not why they can't cross this cell membrane. It's not the force field that rejects them. It's their size. They're just so large. It's like you coming up to a chain link fence. You look at that and you're like, well, there's no way I'm getting through those little gaps. So a big molecule like a glucose or a sucrose is going to get bounced back off the wall of that cell membrane. So very large molecules and anything with a charge, they're both going to get kicked back and won't be able to cross the membrane. But you can see here we have things that do cross the membrane. Things that are able to cross the membrane are things that have no charge on them. So up here we've got oxygen, carbon dioxide, nitrogen, and we've talked about why those don't have a charge on them. And because of that, they don't have any problem. This central part here is not a force field to them. So if you have no charge on you as a molecule, you're going to be able to get right through that cell membrane. If you are a small, so this is where things get a little tricky, because if you have a very large molecule, can't cross. If you have a molecule that has a full charge on it, a full negative, a full positive, can't cross. But then we've got this middle category, and we've got things like water in here. And they are small, which favors getting across, but they're polar, which is like a little bit of a charge. So this force field area should reject things that have a full charge, but they'll kind of let you squeak through if you just have a little bit of a charge on you. So something like water, that's a polar molecule, it's not great that it has a slight charge on it. That's not favored to get across, but it's quite small, so that does favor it to get across. So you can see the arrow here is smaller than this one. That means water has more of a hard time getting directly across the membrane. So what the cell tends to do is it'll make kind of channels for the water to get through so that it can allow water in. So we know that water is able to cross a little bit on its own. cells will typically give water an extra pathway to try to get water in because cells are often very thirsty for water. So we've got this situation where something like water is able to get into the cell. So water is going to see this cell membrane as not really a barrier and will be able to cross. But if you're something like sodium, potassium, chloride ions, you're going to get rejected right at the gate. So you won't be able to cross. So what you have to do with your potato lab is figure out, okay, if I know that my potatoes are made of cells, which means they have cell membranes like this, the solutions that I soaked my potatoes in had both water in them and salt in them. So you have to figure out, based on that, what was going to be able to cross the cell membrane of those potato cells. You've got water and you've got salt to consider. So that's the first thing to think through when you're working on your diffusion lab, is what's going to be able to get into or out of those potato cells and what's not going to be able to cross that membrane. So what's going on when you're looking at one of these diffusion problems is so the first question to ask yourself is there a concentration gradient, right? Is there a place where there's a lot more of something piled up and not a lot of it in another place? And if that's the case, if there's a concentration gradient, then those particles will feel that drive to spread out until they're equal in all places. So the second question you have to ask yourself is, if they do feel that drive, if there is a concentration gradient, are they going to be able to move? So is there a membrane in their way? If there's a membrane in their way, are they physically able to cross that membrane, or are they stuck on one side of it? So what we've got here is a simplified example of a tank of water. There's a membrane in the middle of it. You can see it's a semi-permeable membrane because there are gaps in it, so certain things are able to go through. And what this experimenter has done is they've dropped basically some food coloring on this side of the tank. So we can see these molecules of that dye, of that food coloring. And at first you drop it into only one side of the tank. But what you've got here is a lot of these particles on this side of the tank and none of them over here. So that is a strong concentration gradient. Those particles are going to feel an immediate drive to spread themselves out across the whole tank. So that's what they're going to do as long as they're able to cross. And this particular membrane has gaps that are big enough for these dye particles to get across. So what we see is if we just give them some time, they're naturally going to spread all on their own without you having to force them. They're going to cross over through that membrane. So if you give them enough time, you come back in 15 minutes or so, you'll find that in your tank, that food coloring has spread through all the water in the tank, and it's about the same color everywhere. This is what those particles are going for. They feel this drive because there's a ton of them stacked up over here, none of them over here. Give them some time, and they're going to get to this point where they're spread out equally on both sides. And that's called equilibrium. It doesn't mean that none of them are moving at all. So in fact, if you could animate this screen, what you would see once they've reached equilibrium, same number of particles on the left as on the right, you'll see an occasional particle drift over here, but for everyone that drifts over here, this guy might leave and go back over here. So overall, if you tally up how many particles are on both sides of the membrane, at any given moment, they're going to be equal. But a specific particle might be crossing over at any given moment, and another one will be moving to the other side to take its place. So we refer to that as there being no net movement. So there's no overall, like, oh, they're all moving left to right. There'll just be sort of some random back and forth. But if you were to count up the particles, they're equal on both sides. So that's what equilibrium means. And the particles will tend to keep moving back and forth throughout that situation. So once they've reached equilibrium, there is still movement. It's just not an overall net movement. Down here, we've got a little bit of a more complicated situation. This experimenter has taken our tank of water, put one color dye, one color food coloring on one side of the tank, a different color food coloring on the other side of the tank. The membrane is still there, but there's still gaps in the membrane that are big enough to let the dye molecules go through. So if you look at what's happened here, you'll notice that each color of dye feels its own concentration gradient only. It only cares about itself. So these purple dye molecules are feeling this concentration gradient where they're like, oh, there's a ton of us over here and there's none of us over here. We need to spread out until we're equal everywhere. And that's what the purple dots do. So you can see by the end, we've got the same number of purple dots left and right side. At the same time, the orange dye are feeling their own concentration gradient. So for them, they're like, oh, there's too many of us over here. There's no orange over there. We need to spread out. So if you give them time, they'll do the same thing. They're going to cross that membrane until they are equal on both sides. So the thing to remember when you're looking at a diffusion problem is that every molecule, every atom, every particle, whatever it is, feels its own concentration gradient only. So if you have water's going to have to figure out for itself, like, oh, if there's a lot of water over here, but not a lot of water over there, then I need to spread out until there's equal water molecules everywhere. Salt is going to feel its own concentration gradient. So the sodium is going to feel like, okay, there's a ton of me over here. There's not very much sodium over there. I need to spread out until there's equal amounts of sodium everywhere. So every molecule is feeling its own concentration gradient. Every molecule has to look at the cell membrane and figure out, can I get across that or not? Because it doesn't matter how strong the concentration gradient is if that substance is not able to cross the membrane. So for things to move, for things to diffuse, you need those two factors to be true. You need a concentration gradient that's going to drive those particles to move, but you also need a membrane that's going to let them through. If you don't have a membrane that will let them through, then your particles are going to end up stuck exactly where they are. So what you guys are looking at with the potatoes is you have to figure out what's water going to do and what's salt going to do, because those are the two things you have in this situation. Now, what I'm going to leave you thinking about is this experiment. It's a classic experiment where you have this U-shaped tube and there's water that's been poured into it. And then in the middle here, we've got a semi-permeable membrane. Now, in this case, the semi-permeable membrane works pretty much like a cell membrane. It'll let water through, but it will not let things like salt through. So you can think about these purple dots as salt, as sodium and chloride. So this membrane, you can see the gaps are too small to let these purple dots through. So the purple dots can't go anywhere. So what I want you to think about as you're doing your potato lab and thinking through this diffusion stuff is if you've got all these purple dots over here, and then this number of purple dots over here, what is the situation for these purple dots? Let's think of them as salt. So for these salt particles, are they feeling a concentration gradient? So are they feeling a drive to spread out, move from one place to another place? So that's the first thing to ask. Second thing to ask is, for the salt particles, are they able to cross that membrane? Because if they're not, it doesn't matter what drive they feel. They're stuck where they are. Now, the second thing you have to ask yourself is for the water. So the water is small, so all this pale blue, you have to imagine that that's all made up of individual water molecules. So you can see all this light blue space on the left side. Those are all water molecules filling in all those gaps between the purple. Over on the right side, the light blue is also water. So we've got water molecules filling in all the spaces between these purple particles. So what you need to think about, and I'll leave this for you to puzzle through, what we end up seeing is if this is our starting situation, this U-shaped tube over here, if we let it run, if we just left it on a countertop and came back in 15 minutes to see what had happened, you would see this weird situation where you see the water level over on the left side has dropped, the water level on the right side has gone up, which this situation looks bizarre because it looks like it's defying gravity. So what you're going to be left to think about is, why did that happen? What were the forces driving these things? What was able to move? And how did we end up going from even levels of water over on the left side to just giving it some time, and it seems to magically kind of settle where this side of the water is high and this side is low. So give that some thought, think through it, think through what was going on with your potatoes, and see if you can apply some of these concepts of concentration gradient, diffusion, and the drive to move across a membrane and the ability to move across a membrane that you need to solve out that lab.